Welcome to Spring LIB.
Okay, so we're still talking about inheritance here. And this question starts by wanting us to talk about technology. Okay, and how the theory of inheritance has been supported by technology. And I think the main thing we want to get at here is the karyogram. So the karyogram is basically a series of dyes that allows us to see the alleles being stained, right? And so that's why every time we see a picture of these chromosomes, they have stripes. And so there's a series of different dyes that make the very particular stripes, and we can collect that information on a lot of people and get a sort of standard version. And then once we have that standard version, we can test other people's. And if theirs looks like the standard, we know that there's nothing wrong. And if there's something weird there, then there's a mutation to be aware of. And some mutations are beneficial, some are harmful, and some do nothing. But the karyogram, this series of dyeing and staining chromosomes, has allowed scientists to identify where genes are on chromosomes and also be able to identify when mutations occur. So then we move on to part B, which wants us to connect this theory to Mendel's laws. And the first is the law of segregation. So the law of segregation states that individuals who possess two alleles will pass only one to their offspring, right? So we have two, and the gametes will only take one to pass on. So, and we can see that with this ability to stain, we get very good images of meiosis occurring. And we get to see that, during the different anaphases, when that separation distinctly occurs. The other law we were supposed to be looking at is the law of independent assortment, which we know does not always hold true, right? So this one states that one pair of genes is independent and inherited from another. And so if we're on two different chromosomes, if we're looking at gene 1 and gene 2, then yes, that will absolutely hold true. But if we're on the same chromosome and kind of close together, then it might not actually be true completely. We will get linkage occurring. But we can also view that, and so that's how we know. We can say whether or not independent assortment will take a role, because thanks to the karyogram, we can see exactly which chromosome the gene of question is on. So this is a quicker one, more words than pictures, but hopefully that all made sense.